Okay, welcome back to our second part of neurobiology. We're going to spend today talking all things brain and all important bits of the structure, the function, specialized locations, everything to do, everything you've ever wanted to learn about the brain. So to start with, we're going to talk about the functions. So I'm just going to throw up all the labels here, but what would be really good for you to do is to maybe pause this before and make sure that you can label and give the functions of all of these parts. So to start with, we're going to start at the spinal cord, which is just where nerve impulses happen, and as you move up you hit the medulla oblongata, which is important for unconscious processes like breathing, heart rate, stuff like that. As you move up, right at the top of that is the hypothalamus, which regulates like homeostasis, but also different hormonal secretions. To the front of that is the pituitary gland, which is like really important for hormonal control, and behind the hypothalamus is the pineal gland, which deals with like your internal clock, your sleep and wake patterns. Behind all of this and your spinal cord and all of this inner bits is the cerebellum, which coordinates movement and balance, and then all of this other stuff, which is like the pink part of your brain, is your cerebral hemispheres, which is important for complex thought, memory, basically everything else. Your cerebral hemisphere can also be broken down into 4 lobes. You have the frontal, parietal, occipital, and temporal. The frontal is where your forehead is, so that's kind of like all your problem solving, movement, motivation, all of that. If you move directly back on the top of your head, you would hit the parietal, which is your sensory input. And then below your parietal, where like your ears are, is your temporal, which deals with auditory, which makes sense because it's right near your ears. And then all the way in the back is your occipital, which deals with visual. So with the brain, how we learn about its functions we kind of learn about the functions in 3 different distinct ways. You have the MRI, lesions, and animal experimentation. And I'm going to go into each of those a little bit more. So our first one is the MRI, which is really all about determining areas of the brain that are stimulated with blood, because oxygenated hemoglobin and deoxygenated hemoglobin respond differently to a magnetic field. So you can see where blood is flowing to, and then once the oxygen gets dropped off, it no longer portrays itself on the MRI. And you can see this by different colors. And so as things are happening in your brain, right, you could show people pictures and see what parts of their brain light up. You could test this on people who are addicted to drugs and see what part of their brain is no longer functioning in the same way. So this is used just really to determine where brain function is happening. However, not all brain activity is detected by this, so like while it's really important to use, it's not the only thing, the end-all be-all of determining brain function. There's also lesions, which are sort of like cuts, right? So abnormal areas in brain tissue, this could be from accidents or it can be from diseases, which would indicate what happens and the effect of the loss of an area. So an example of this is aphasia, where if you have a specific damage to the left hemisphere, you then maybe have trouble understanding language. So one example is like just an abnormal area. A second example is if it's specifically in the corpus callosum, which is the really thick band that connects the left and right hemisphere. So you could cut that band, and sometimes it's done for a reason, like to relieve seizures, and then other times it's done or has been, it exists, and it can lead to split brain personality. It can also be used to determine the roles of the left and right hemisphere, right, to see, which parts of like language understanding or language acquisition happen on the left side versus the right side. And then what's cool about this, though, is your neurons basically can reroute anything. if they're young enough. That's the key, if they're young enough. And so even though you may have a lesion somewhere in your brain, your neurons can reroute the function to a different part of your brain. So lesions do affect brain function, but it's limited because functions are usually involved in multiple areas and neurons can reroute the information. So very interesting. And then last is animal experimentation, which is done by stimulating a region with electrodes or doing a lobotomy. They're highly invasive, potentially damaging. So although animal models is frequent, like using animals for experimentation is frequent, it's not really, it's sort of frowned upon. A lot of scientists want to use primates because it's the best comparison to humans, but ethically you have to take into consideration that that's like really not okay. And of course you're limited by the fact that brains may not be so similar, and you know, you can't really make a valid comparison. Additionally, some people do autopsies, or like when you donate your brain to science just to see some other functions of the brain. The next part that we're going to dive into is the nervous system. So the nervous system is broken into two parts. There's central and then peripheral. The central is like pretty straightforward. It's the brain and the spinal cord, the main players in the nervous system. And then peripheral is basically everything else. So any nerve that comes off of your spinal cord is part of the peripheral nervous system. And from there, it can be even broken up more. So it can be broken into sensory versus motor, which is like pretty straightforward. Sensory is like sensing something is soft, and motor is like everything else. And then from motor pathways, it can be broken into voluntary versus involuntary movements. And then the involuntary is broken up into fight or flight or rest and digest, which is the sympathetic and parasympathetic division. To bring that even further, you can look at this, which I'm not going to go through, but if you wanted to pause this, you could look at it yourself. But these are the differences between the two involuntary versions of the nervous system. And then to get into death itself, so we define death as four pretty distinct ways. The first is probably what you're thinking of when I say death, which is biological, which is the cessation of life of the whole organism. Like it's nothing is working anymore, and that's biological death. Cardiopulmonary is when only the heart and lungs cease to function. Right? Cardio, heart, pulmonary, lungs, only those two cease to function. Whole brain standard is when all parts of the brain are non-functional, including the brainstem. And this is what happens when individuals are kept in a vegetative state. You would check this using the pupillary reflex, which I'm going to talk about on the next slide. And the fourth definition of death is higher brain standard, which is very similar to whole brain standard, except the brainstem is still functioning, so homeostasis is still being controlled through the brain. I was talking about the pupillary reflex on the last slide, but basically this is just when you shine a light in someone's eye, right, your pupil gets smaller, and then when it's darker out, it gets lighter, and this is due to the photoreceptors in the eye. And what happens is you get an impulse, and it travels through the optic nerve to the medulla oblongata, and then back, and from there, the muscles either constrict or dilate, and if your brain is working, then you would have a reflex happen. Now, we're going to move a little bit in a different direction. This goes sort of all over the place, but I want to make sure that I'm covering all the basic overviews of the brain. So we're going to talk about the cerebral cortex. Now, only mammals have this, and the increase in the human cerebral cortex is partly due to thickness, but more due to increased folding. And you can see that in between the rat, cat, and human, that there's so much more folding in the human brain, and this leads to more surface area, and that's the most important piece. The folding and the surface area, and that's why humans are more intelligent than cats, and cats are more intelligent than rats, because you can see the folding in the 3 different brains and the differences between them. Now, the cerebral hemispheres is this is like the different lobes in your brain, more than half of the brain mass, and there are 2 different kind of vocab words, I guess. There's the gyri, which is the ridges and the bumps like on the brain, and then the sulci, which is the grooves in between the gyri, so the inner parts. So between the gyri and the sulci, this is what gives you the surface area that is lending itself to the intelligence. Now, these next 2 slides deal with brain size versus body size, and for every species there is a brain-to-body ratio. However, it doesn't really mean anything, right? Because humans have a 1:40, meaning that their brain is 1/40th of their body. But so do mice. And that doesn't mean that we're the same. And birds have a higher ratio. But are birds more intelligent than humans? Well, crows are pretty smart, but I wouldn't say that they are smarter than humans. So although we can have this ratio, it doesn't really tell us anything. But we can take that and put it into this encephalization quotient, and then kind of get a slightly better idea of what it means in regards to brain mass based on an animal size. So there's this formula here, and it basically takes the weight of the brain, the weight of the body, there's a constant for each species and an exponential constant, and you do all of this math, and it gives you this quotient, right? So you have the we're going to look at dolphin and chimpanzee. So dolphins have 5.31 EQ, and chimpanzees have a 2.49 EQ. And what that means is that a dolphin is twice as encephalized. I don't know why I struggled so much with that word. And basically what that means is that the ratio, what encephalized means is the ratio between the actual brain size and predicted brain mass for an animal of that given size. So the dolphin is twice as much encephalized as a chimpanzee based on their EQ. Okay, and you can see that because 2.49 is about half of 5.31. So based on their size, this is the number that they would get. You don't really need to know much about this except that body size to brain weight doesn't really have a big correlation. So there are 3 specialized areas in the brain that we're going to talk about. 2 of them deal with language, and 1 of them deals with pleasure. So Broca's area and Wernicke's area both deal with language. Broca's is the production of speech, so that means that an injury here means that you can understand it, but you can't maybe talk. And Wernicke's is the opposite, so it controls speech comprehension. Injury here means that you can talk, but maybe not understand speech. And then the nucleus accumbens is the pleasure-reward center of the brain. So this is where the release of dopamine happens. This is where like all of your feel-good emotions happen. This is like your reward center. And then, if you didn't know this, sensory input and motor control is done by the hemispheres. So the left hemisphere receives sensory input from the right side of the body and the right side of the visual field and controls muscle activity in the right side, whereas the right hemisphere controls all of the left side. So it does the opposite side, right? Left controls right, and then right controls left. The homunculus is a model. You should definitely be aware of what homunculus is, and it shows the relative space that a human body parts occupy on the somatosensory cortex and motor cortex. And basically what this shows you is the importance of different body parts. Like you can see here that the face takes up a huge amount of space, but your abdominal area takes up barely any. So it just kind of shows you what's important and what's not in relation to functions of your brain. And then our last slide here is talking about metabolism. So neurons are your brain cells, and they require a ton of energy to set up resting membrane potentials to constantly be able to do whatever it is that you want them to do, right? Neurons are responsible for sending every single piece of information out to your body to do whatever it is that you want to do, right? If I want to write my name, neurons are responsible for getting that output sent to them. So that means it's a ton of energy. And most vertebrates use less than 10% of the energy consumed, right? We've learned that before. And adult human brains use over 20% of total energy consumed. So, just on their brain. So this is like a huge energy drain. So this is why if you're like really tired or you haven't eaten in like a really long time, your brain gets like all fuzzy because it just can't keep up because you're not giving it enough energy. So this is my final thing for today, but also go eat a snack because your brain needs it. Your brain needs all that energy to understand all this good stuff. Okay? I'll see you next time.